Thanks for the introduction, guys. Let's go ahead and share from here. Um, yeah, thank you. Thanks so much for the invitation to speak. I think this is a neat piece of work. We had a lot of fun writing this, and um, it was I'll give credit to where credit is due to Dr. Joyce Kang. She's an MD PhD program, um, and she's in the Harvard Big program. She really did all of most of the work to see this through and to make sure that we test it in as many different kind of conditions as we can. And thank you, Lang and Daniel and the organizing committee of this for inviting me. And I really encourage that if you have any questions during the talk, just jump in, and we can have a discussion as we go. Lang, am I gonna be able to see or hear people as questions come in, or
Yeah,
you'll
so
moderate?
people's I can monitor
Okay,
that and I'll let you
okay,
know if there's any question from the Q&A. So the
okay,
audience are welcome to type your question in the Q&A so we can talk.
excellent. All right, cool. Um, yeah, so let's get started. So the work so first I'm gonna get financial disclosures out of the way. Some places my lab gets funding from. I also do consulting for Mestech Therapeutics. So today the work that I'm gonna present is basically this paper, and we're gonna walk through the background, the motivation, and some of the key results for this paper. And this paper really describes this Symphony algorithm for doing single cell reference atlas mapping. So that whole thing will motivate and describe why it's useful, where it comes from, and what we can use it for. And yep, so Joyce Kang, a graduate student in Shomari Chowdhury's lab, really drove this project from the beginning and drove it home. And I started this project when I was a postdoc with Dr. Shomari Chowdhury here in Brigham Genetics, and we finished it when I started my lab here in Brigham Genetics. So thank you to Shomo and his whole lab, everyone who contributed there. Okay, so really, really big picture, all of single cell data. There's a really big promise, right, of understanding biology and medicine through the lens of really, really finely phenotyped cells and cell types, and the idea that we can phenotype cells. We can understand their lineage and their function through the expression of their genes. Is really where this whole field, the idea that we can do this, and the idea that we can do this with millions of cells at once, and across all organ systems in the human body, this really leads to the promise that we can sequence the transcriptome of every single cell type that exists in the human body. And these are projects like the Human Cell Atlas, and projects like the NIH funded HubMap project are really built around this. Really built around the idea that we can, by assembling experts in all these different organ systems, and leveraging these great technologies that we have to do single cell genomics, we can profile the whole thing, and we can create unified and comprehensive atlases of the cell of all the cells in the body, just like we have a unified and maybe not so comprehensive but approaching comprehensive view collection of all the genes in the human genome. So this is the grand promise, right? Is to be able to build this huge unified atlas of all cell types across all tissues, and eventually across all diseases. And the data has been coming in for years, right? So I think since 5 years ago, I didn't include the graph here, but we were sequencing hundreds of cells at a time, and now it's not unthinkable to do an analysis with 1 billion cells in a few years. We're regularly doing analyses with millions of cells. And so the data is coming in, the data is there. The problem is that it's not coming in the most uniform way. It's not like there's one lab that's taking, all cells from multiple organs. There are some exceptions, but I say for the most part, there are not, labs that are just taking all organs, from the same person and running them in a super uniform way that gives us one view, one shot experiment kind of experimental view of all the cells. Instead, what's happening is that multiple labs, right, each one an expert in their own organ system, each one an expert in their field, is generating these very high throughput data sets of millions of cells from multiple donors. And so what we have is this really piecemeal thing where data sets are coming from different donors, different organs, different labs, sometimes different technologies, very often different tissue disaggregation protocols. And so what we get is, instead of one easily interpretable unified atlas, we get kind of a blob of data, right? And in order to be able to make sense of this big heterogeneous blob of data, we need to be able to map it into some kind of unified space where at least we know that if these cells, are in this region, they're T cells, and if these are in this region, they are hepatocytes, right? We want to be able to have that kind of definitive knowledge, right? And so the first thing that the field really grappled with, and could say is still grappling with, and still grappling with some of the nuances, but the thing that everyone really had to face like 5 years ago was, how do we take all of these diverse data sets and blop them into one thing, right? And I'm giving you kind of a very almost like a worst case scenario, right, of multiple donors, multiple labs, multiple organs. But in our practice, even if we take blood from 5 different donors and we run each person's PBMCs in a different single cell experiment, we're going to see massive batch effects. We're going to see the cells really grouping by donor a lot of the times rather than by inherent cell type, right? And so at all levels of single cell analysis, big or small, as long as we have multiple experiments, as long as everything was not run in one batch, one experiment, we have to do some kind of integration. And we contributed to this field with the Harmony algorithm in 2019. And we really try to cater to all scales of this, right? Whether I have 3 donors that I ran and I want to integrate the batch, or if I really do have 1 billion cells, 10,000 data sets, and multiple organs, multiple labs, multiple donors, how do we integrate all of those factors into one thing? That's what we're really trying to design this algorithm for. But I'm not going to talk about this one today a lot, but it is important, I think, to understand that this is the beginning of how we're thinking about it. And so this is what Harmony lets us do, right? Let's just take cells that are in, this is an example from the paper, of pancreatic islet cells from 5 different technologies, from 60 different donors. And if we just do a naive analysis, we don't get cell types lining up with cell types. I think people if you've analyzed single cell data before, this is not a surprise. If you haven't, I'm sorry. This is kind of normal, that things will not group by cell type automatically like this a lot of times. And so, with Harmony, we model the effective technology, we model the effective donor, and we remove it in order to identify shared cell types across donors and technologies. But what we get is one shared unified space where we can map all 60 donors, all 5 technologies into, right? And so this is kind of the difference between an agglomeration of data sets and an actual atlas, right? Is where things share a unified space. that is biologically meaningful, and we can make do biological inference in that space. So then the question is, let's say that is, let's say we've 80% towards solving that issue. How do you go from 1,000 independent data sets to a unified atlas? How do we use them, right? Because this is what we really want to do, right? And so to make an analogy to genetics, there is a human genome, and massive amounts of effort went into creating the first draft of the human genome, and now we've created, we're people are still working on making better and better and better reference genomes, right, to understand where all the genes are in the genome. But the real power is not just having that atlas and browsing it, right? The real power is, now I did my own experiment. I sequenced a new person in a new condition, and I can map those reads from that person into the reference genome, and I know exactly where they fit, and I can compare it to the reference genome and say, okay, there's usually 99% of people have a G in this spot, but my new data, there's a T there. What does that mean? Is there an association, right? And so by comparing against a reference, I can take a set of reads that's kind of hard to interpret, but in the context of the reference genome, I can make inferences. And so this idea, that we didn't invent, a lot of people are really thinking this way, is how do we, once we have an established harmonized reference atlas of single cells, how do we then map our new single cells into that atlas and make inferences about our new cell data set? And so this is the analogy of everything, right? So instead of having reference genome, we have a harmonized atlas. Instead of having a set of reads like in genetics, we have just raw single cell RNA-seq profiles, or any kind of single cell genomics profiles of your cells. Instead of a mapping algorithm, we have our Symphony algorithm, which is a single cell reference mapping. And instead of inferring genetic variants, we're going to infer other things that are encoded in the harmonized cell atlas, such as cell type. Multimodal expression will go into that. Disease associations will go into that too. So we can transfer information over from the reference atlas into our new data by doing this mapping. This is the plan. This is the hope. And the way that we view this problem is that it's kind of a special case of integration. So in single cell data integration, which Harmony addresses, we have 1,000 different, or in this case, we have 5 different data sets, and we don't have a unified reference frame for them, but we want to kind of combine them all, and they're all equally important, and we want to combine all of them into one unified reference frame. In reference mapping, we already have a frame of reference, and it came from these 5 previous data sets. Now what we want to do is we want to map a 6th one in, right? So what we could have done is we could have just said, okay, well, let's reharmonize all 6 data sets. But it's really slow, it's not feasible. Sometimes these reference atlases are really, 10 million cells. Not everyone has the power or the time or the interest to reharmonize everything. And also, sometimes I want to keep my reference atlas intact. I don't want to reharmonize, reintegrate, recluster, reannotate, everything. I just want to say, where do these new cells fit into my existing frame of reference, right? So it's an integration problem, but it's an integration problem where one data set is static, and the second data set is the one that we want to integrate into the first one, rather than we have a bunch of equal data sets and we want to integrate all of them. Okay, so how did we address this? So this is what we want to do. This is a special type of integration algorithm, basically. And so this is how Symphony works. First, we build a reference atlas, a harmonized reference atlas. And so this involves the standard harmony workflow. And then in the middle here, after you've done PCA harmony, clustering, trajectory analysis, whatever you want to do, you've annotated every single cell, right? So every cell has maybe a cell type, maybe an association with disease, maybe a position along a developmental trajectory. It has some kind of annotation that you've done, that you've annotated that cell with through your own analysis. And then we take that reference atlas and we compress it. And we say, what's the minimum? We keep only the minimum pieces of information that we need in order to map new data into that reference. Then the second part is, once we've harmonized an atlas, created an atlas, compressed the atlas, then we do the Symphony mapping. We take a new data set, we map it into the same low dimensional space using saved PCA loadings. And then we basically do kind of the second half of harmony, which is clustering. Familiar with harmony, clustering and correcting, but only correcting the new query cells. And then in panel D, after you've done this, the new query cells now have locations within the same frame of reference as the atlas, and we can make comparisons. We can say, okay, this unannotated query cell, who is it next to? It's next to all of its 10 neighbors are all T cells. It's also probably a T cell. It's in a region where I only had cancer cells. Okay, maybe the cell is somehow relevant to cancer also, right? And so we can do these kind of annotation transfers once we've embedded the query into the reference. Yeah, and so yeah, we really want to strike.
Sorry, can I ask one question here?
Yeah, Daniel, please.
So
or Liang.
Yeah, that's fine. I think Danny maybe also have a question. I saw that he actually raised the hand. For cells which cannot map to the reference, how are we going to do that? Can you move to the next? Oh, okay. Are you going to talk about that?
Not as much actually. So it's a good point to talk about now. We do address that in the paper in one of the supplementary analyses. So definitely important, right? So this is a situation we've considered, right? So you can say that you have a data set, your reference data set for some reason is only T cells and monocytes, and you want to map B cells into there, right?
Right.
Yeah. So we've tested that, and there's a gradient, right? There's a continuous thing, right? So if I only have T sites and monocytes and I want to map B cells into there, I will actually successfully get B cells. I will be able to. We have a metric that Joyce developed to be able to say that, okay, these cells actually don't look like anything in my reference, so I'm going to kind of keep them separate. But you can imagine that this is kind of a tricky problem when I have, I don't know, TPH cells in my reference, but I map TFH cells, which, in immunology, these cells, in terms of surface markers, only have, one is CXCR5 positive, one is CXCR negative, and so, in that case, probably the TFH cells are going to map into the TPH cluster, and maybe Symphony will not catch the fact that there's actually a super subtle difference between their gene expression, right?
Right.
So I think it's a matter of size, a matter of difference and gradation. Yeah.
Thank you. Daniel, you have a question, right?
That covered my question. Thank you so much.
Oh, that covered. Okay. Good. Okay, cool. So we have this algorithm Symphony. We build a harmonized reference. We compress it, and then we can map new cells into it. Okay. So this is that was figure 1 in the paper. So this is figure 2 in the paper. So figure 2, we really want to emphasize the relationship between harmony and symphony. So in panel B on the right, what you're seeing is, so first of all, the data, right? So this is 3 data sets of PBMCs from healthy blood donors, all done with different 10x platforms, but using different 10x library prep kits. So one is using 5', one is using 3' version 1, one is using 3' version 2 libraries. And in panel B on the right, what you see is the result of harmonizing all 3 together. So it's not using Symphony, it's just using harmony to integrate the 3 into the same space. And at the top, you see the cells are colored by the data set of origin. On the bottom, they're colored by their previously annotated cell type. And in Symphony, what we want to make sure is that if we build a reference out of 2 data sets and then map the 3rd one in, that we should be able to get similar results to what we got when we just did harmony on 3 data sets. And this is what we really confirmed. We really do get very comparable results with a few minor differences. Some CD4 T cells, group with some CD cells. But even in the harmonized version, they're so close to each other that these are kind of cells that can move back and forth, are difficult to annotate definitively. What's different between panel A and B, though, is the actual UMAP itself, if you notice, right? So in panel B, the UMAP is learned on the embedding of all 3 together, whereas in panel A, the UMAP is learned on just the joint embedding, the harmonized reference of 2 data sets, right? And so when we map the 3rd data set in, it doesn't change the UMAP embedding because it doesn't change the reference embeddings. And so this is what Symphony achieves, is it keeps the underlying reference atlas stable, and then allows you to map the query in to see where it lands in that space. So this is the without disturbing part. The quick part is also kind of important, right? So to make this a really usable tool, we made sure that it's scalable and efficient. These are some experiments that we did to see just how scalable and efficient it is. So this is a data set from should have put the reference here from blood cells, but we don't do much interpretation here, so I'm not going to describe the data set, only to say that it's a pretty large data set, or was considering 500,000 cells from over 60 donors. So it's large both in the number of cells and in the number of data sets, number of donors here. And so if we look at the 1st and 3rd panels, we see that, remember, Symphony works in 2 parts, right? Reference building and then query mapping. So the reference building part of Symphony pretty much is the same as doing harmony, right? And because that's really the rate limiting step of reference building in Symphony. And the time really does get, pretty sizable, it gets sizable once you reach big numbers, right? So still doable, if you need to do it, 10,000 seconds, I don't know how many minutes or hours that is. But on the biggest data set, it takes about 10,000 seconds to integrate, 64 donors, and 500,000 cells. And then the compression part is really fast. So this is the part that does take time. But the nice thing is that in most cases, you really need to only do that once. And in fact, in a lot of cases, once this stuff kind of becomes more mainstream, you never have to really build it. You just get a reference data set from somebody else. So you read a paper, you say, oh, that's a really cool analysis they did. I'm not going to download their data directly. I'm going to download their ref atlas, and I never have to analyze 1 million or 10 million cells myself. I just download the compressed reference atlas. Then I can take my own data and map it against their reference atlas, and that's the query mapping part, and that's the part that's super fast. So it's the part that's super fast, and it's totally independent of how big the reference atlas is, because the reference atlas is compressed already. And we see the biggest cases that we tested, we tested mapping 100,000 cells from 64 different donors. And it still takes about, less than a minute, on a standard machine, to map all of these cells. Whereas analyzing 100,000 cells from 64 donors would definitely, all the pre-processing steps alone would take much more than a minute. It would take hours to kind of go through and days to interpret. So this is an important thing to know, is the efficiency of Symphony really comes in remapping. And if you already built an atlas, this is a best case scenario. So next, what I'm going to go through is walk you through the rest of the figures in the paper, and the rest of the figures really highlight different aspects of Symphony and different use cases of Symphony. We'll start with the really, really simple one, which is basically the one that we already walked through, right? Which is, I have a reference built out of some number of data sets. There's cell types that I've annotated my reference cells with, and all I want to do is take some new data set that's same tissue, same kind of thing, and it's just one data set, and I want to pop it into the reference and then predict the cell types, right? And so this is the easiest case, and this is what we did here, right? And we predict cell types based on nearest neighbor classifiers. The more complex case comes when we have multifactorial design. So in the reference, but especially in the query, this is where it's really interesting. So in the query, I don't just have one data set that I ran and I want to see what the cell types are. Maybe I have a query that itself is a study, right? So in this case, the scenario that we've created is that this query has both human and mouse data. The technology in the query is different from the technologies used to build the reference. And I have multiple biological replicates within mouse and human subsets of my study, right? So I have multiple levels of variation that I would want to get rid of in order to identify cell types and be able to map it back into the reference. So in Harmony, if we were to analyze all of this together, what we would say is put all of these cells and genes into one big cell by gene matrix, and then do PCA on the whole thing, do Harmony on the whole thing, and when you do Harmony, correct for both species and donor and technology, right? In Harmony, you can give more than one batch variable, so you can give it 3 batch variables, and Harmony would take care of removing the species, donor, and technology effect. In Symphony, it's something similar. What we do is that when we map the query, we tell Symphony for each cell which species it came from and which cell which donor each cell came from. And Symphony then does 2 things automatically. It both projects the query cells into the reference, and it simultaneously does batch correct in the query while it's doing the mapping. And so as a result, what we get is 2 things. First of all, the query cells are mapped correctly where they should be in UMAP space. They cluster with the correct cell types. But potentially equally as important is that if we look at the color the cells by donor, so the different shades of blue here are different human donors, different shades of red here are different mouse replicates. We see that they're very well mixed, right? And if they didn't mix, you could pretty easily show that the reference mapping would also fail. So this is kind of a situation where I'm not just going to map one data set at a time. That would be really tedious. I'm going to take my whole data set that I've generated, map the whole thing, and simultaneously do reference mapping and correct for batch effect in my query. So this is, especially for those of us who work with, replicates or human data, from multiple studies, this is pretty important. Another thing is that sometimes we're not just interested in cell type. We're interested in things in non-discrete annotations, right? And so that's why we included this this data set. I think this is fetal liver development, largely fetal liver hematopoiesis. Comes from a paper from a few years ago, and the reference has mouse liver hematopoiesis cells, hematopoietic cells, and because it is an embryonic system, and it's capturing a system where cells are dividing and differentiating, what we see is not discrete terminally differentiated cell types, but instead what we see is continuous differentiating gradients, developmental gradients, right? So there's an HSC cluster in the middle somewhere. There's lymphocyte progenitors, there's erythroid progenitors, there's early erythroid cells, mid erythroid cells, late erythroid cells. And we had to kind of label them for convenience, but really they exist along a smooth continuous gradient. And so, in our reference data set, we can make a UMAP just like we did, but we can also do other embeddings. So I think here we decided to go with a force-directed graph layout, which is potentially, better for visualizing these smooth branching trajectories. And when we do our query mapping, the only thing we learn about, when Symphony does the query mapping, is what are the harmonized PCA embeddings, right? And so this does, Symphony itself does not depend on UMAP or any other low dimensional embeddings other than the harmonized PCA embeddings. And so what we did was, in addition to predicting the UMAP location of every query cell, what we did is we predicted the force-directed graph location of every query cell. And to make sure that we kind of could interpret things, in our query data set, we limited ourselves to this one developmental branching event, where progenitors differentiate into mast cells, megakaryocytes, and erythroid cells. And what we're seeing here is these query cells as they're mapped into this trajectory, into this two-dimensional trajectory representation. And what's really cool is that we can really see these branching events the way that we expect them to happen, which is really the power of having a good reference that's complete, and that has all these intermediate stages, and a good algorithm like force-directed graph that can represent these branching events and these smooth trajectories. And all Symphony does is really just, map these cells into that same space and leverage that same high quality reference model in order to say something about these query cells. And then once we've mapped these query cells into the reference, of course, we labeled them with these discrete labels, and we can see that there's a little bit of smoothness and ambiguity between like early erythroid and mid erythroid cells, which is totally predicted, all right, because this is really not discrete clusters, this is a trajectory. And so we went ahead and looked at some of these lineage marker genes for erythroid cells, for megakaryocytes, and for mast cells. And we can see not only are these lineage marker genes the highest at the end of these trajectories, which we expect, these are the most differentiated cells, but we also see these really nice smooth gradients that we don't lose when we do the reference mapping. So if you have a developmental system that you're interested in, or if you have anything that's really more trajectory than discrete clusters, Symphony still works for you, as long as your reference models the trajectory well. Another use case that's especially important for those of us working in the hospital settings with patient samples is disease and healthy, right? So one of these big promises of single cell is that we can have a giant complete atlas of healthy cell types, and then we map disease cell types in, and we can see what's different, what's changed, right? So we have a healthy background, a diseased foreground, and then we can say what's different about the disease cell states, and some of them will really pop out, and we'll say, aha, these cell types, these are the ones that are relevant to disease, because they don't look like the background, like the healthy background. So Joyce found this. She constructed this new example where she took a healthy fetal kidney reference with annotated cell types and mapped renal cell carcinoma cells against this reference. And what we saw is that we pretty much got the most of the cell types right, and that's fine. That's what we've been doing in every in all of the benchmarks. But what's really interesting is when we went in and asked which cell types mapped well and which cell types did not map so well, which cell types looked different from the reference, and that's really figure E. And so this is a mapping metric that Joyce developed, and the lower the mapping metric, the more similar that cell is to the reference, and the higher the metric, the more dissimilar it is, the more novel it is compared to the reference. And so we can see in this cancer sample, NKT cells, Tregs, B cells, NK cells, they kind of looked similar enough in the fetal reference and the cancer sample. The immune cells were pretty similar. As we go up the chain, we start seeing tumor-associated macrophages are less similar. Endothelial cells and fibroblasts, which are not immune cells, right? But they're stromal cells that are going to be different in fetal tissue versus cancer tissue, have higher scores. And then these cell types that are colored in red here, TP1, TP2, cycling tumor especially, are the ones that really did not map well at all to the reference. And so these are the ones where there was no good analog in the reference atlas. And so if we were to do this without any kind of knowledge, we would Symphony mapping would really tell us, hey, pay attention to these cells, because we think these cells are the ones that are different in your sample when we compare it to the healthy fetal kidney reference. So this is how we think about the disease and healthy. Finally, there's this really exciting thing of multimodal data sets now, right? When we started Harmony and Symphony, we were really thinking about, we have a single cell RNA-seq reference and we have a single cell RNA-seq query, and we want to transfer, we want to label cells with cell type, with differentiation, trajectory, position, whatever. But then these technologies started evolving really quickly, where in the same cell you can measure multiple things. Now you can measure RNA abundance and chromatin openness, right, at the same time. Now you can measure RNA transcript abundance and spatial location at the same time. Or in this case, this is the multimodal data set that we used for our example. We're measuring RNA-seq and surface protein expression at the same time. I think this panel is somewhere about 100 different, primarily T-cell related surface marker proteins that help us phenotype the T-cells really well. And so here, the way that we analyze this reference is going to be really different, or it's not going to be really different, but it's not going to be our standard Harmony pipeline, right? Instead, what we do is we want to not just run all the genes and all the proteins through one PCA analysis, although you could. What was really interesting to the authors here who published this data set was to find the overlap between the variation in the protein expression and the variation in the gene expression. So instead of using PCA to construct the reference, instead what they did was they used canonical correlation analysis. which is a nice tool to find a low-dimensional embedding that basically finds the intersection between the variation in the genes and the variation in the proteins. And so what you're seeing here is a UMAP that resulted from doing CCA on the gene expression and protein levels, and then doing a UMAP on I think the RNA, the embeddings, the CCA embeddings for the RNA side of the data. So what's cool about this data? What's cool about this data is that the reference has two things that are measured, mRNA and protein, but the query doesn't have to. The query can have mRNA only or protein only, and the Symphony approach still works. In fact, it's even potentially more interesting, right? Because in this case, if our reference has mRNA and surface protein, but our query only has mRNA, what we can do is map the query cells into the reference and infer what protein expression they have. So to test this, what we did was kind of a cross-validation approach. We split this giant data set into a reference and query sets, and we used both mRNA and protein expression to build the reference, but we hid the protein expression from ourselves for the query cells. And then we asked, if we only use the gene expression to map the query cells into the reference, can we correctly predict gene expression in that test query set? This is the results on all of the I guess it's not 100 genes anyway, but on the protein expression. So this is for each of these genes. It's the correlation between the true measured protein expression and the protein expression that we predict with Symphony. And so for most of the Pearson correlation is something close to 1. It falls down to around 0.8 for some of them. And typically these are lower expressed genes or lower expressed proteins, let me say, lower expressed proteins. And so this is, for some of the well and not so well correlated proteins, this is the true expression that we know that we hid from ourselves, and then the predicted expression on the right. So for CD4, you can see that you really can't distinguish the two, right? And that's what we see in the bar graph there. There's like a correlation of 1. CD69, visually, there's not too much difference, but when we go in and fine grain, there is some difference in intensities. And so we think that this approach works pretty well to be able to have a multimodal dictionary. I think it's another word people use sometimes, and then be able to map unimodal data into a multimodal reference, and then learn something about protein expression, spatial location, epigenetic openness, anything else that's in the reference but not in the query. So I'll end with that. I'm going to thank everybody. The software is on GitHub. You can install it through CRAN in R. There's vignettes, there's tutorials, there's README files, there's pre-built references if you're interested in trying some, and of course issues if there are any issues, and we'll try to do our best to address it. And yeah, my lab is also still growing. We're definitely interested in this kind of work. We're funded by the Chan Zuckerberg Initiative to build Harmony for a billion cells. And so we're definitely keeping that direction strong. And we're also involved in doing a lot of work with spatial, single molecule, spatial technologies. So if any of that is interesting to you, please drop me a line and we can talk. All right. Biju has a question.
Yes. Let me just unmute Biju.
Yep.
Okay, Biju, you can talk now.
Yeah, thank you. Thank you, Dr. Ilya, for a nice talk. I was intrigued by the talk on like the where you are trying to do Symphony on the healthy and the disease model, and something that we are also trying to work on. And I was wondering, like, when you are building this reference atlas, are you only having healthy cell types, or are you also trying to build disease models like some cancer types?
Yeah, I'll give you a short answer and a practical answer and a philosophical answer. The practical answer is you can, but if you can also include disease in your reference, why not? Right? It's even better. And then maybe that's both the philosophical and the practical is, we kind of said this is a proof of principle of if you just don't have access to diseased samples in your reference, that can happen for a lot of reasons. Maybe you're studying a rare disease, maybe you're studying liver and 99% of the world studies one disease in liver, but you study another one, then it's cool because you have new data set, right? But it kind of sucks because you can't directly compare your cells to other cells from your disease. So there's practical limitations. But if you're building your own reference and you can include, multiple donors, multiple diseases, or cells from the disease you're interested in, why not? You're only gonna make your life easier if you do it.
Okay. Second part, which is like a technical question, like how memory intensive or computation intensive is Symphony?
Yeah, it's really lightweight. I only put runtime here, but the memory, I think we put memory somewhere in the paper too. The most intensive part is this one is taking, the time and memory intensive part is building the reference. You can't really, the most memory intensive part of Harmony is doing PCA. So if you can do PCA, that's after that it's a little bit memory wise, it's better after that. But the actual mapping part is super fast. The more things you map, obviously the slower it's going to be. But it's it's pretty lightweight if you're only using especially the compressed version of the reference.
Okay.
Yeah. Hope that helps.
Thank you.
Mm-hmm.
We have another question from Ashish. So Ashish,
okay.
I just unmute you.
Hey, hi, doctor. Thank you for the talk. So I have one question related to what Biju asked. So while creating the reference from the cancer samples, like if we have some cancer samples which don't have like a certain cell types as the kind of other samples, so how do we actually take care of that? So
in the reference,
so basically there can be a case in like which one patient have more than one type of cancer. So if we take that sample and if we put it in the whole thing, so how are we gonna get the reference?
It depends on the biology, I guess. It depends on how different they are, really, right? Like we saw in that healthy diseased one, right? Probably the T cells across different cancers. Maybe a cancer immunologist will tell me I'm wrong, but I think I've looked at immunotherapy responses in like 4 different cancers, and the T cells look pretty similar across different cancers in different patients. I think the similar ones are relevant to disease. Epithelial cells, malignant cells are obviously more different, as when you look across tissues and across donors and across tumors. So when building the reference, I guess this is more of a Harmony question, because this is not the mapping. But I think in Harmony, again, it's kind of a matter of degree. If the malignant cells from one patient are so different from the cells in the other 10 patients, Harmony should be able to leave them alone and not integrate them. They should be able to leave them in their own cluster. But if they are different but not different enough, then Harmony will say, okay, the biology is 100% confounded with batch. So, until you give me more replicates, I'm gonna merge them.
Okay.
That's kind of the approach.
Okay, thank you.
Or that's what happens.
Okay, thank you.
Great. Any more question? I actually have one question. So does Symphony can be used to spatial spatial transcriptomics data also?
Yeah, in theory. In practice, it's tricky. I would try it yourself and see what happens. So, okay, so here's my 20-second thing about spatial transcriptomics data. I think if the cells are really, really cleanly segmented and you have very accurate segmentation, I think you won't be able to tell the difference between single cell and spatial data. And I guess we're talking about like MERFISH, CNEUM, COSMICS here, right? We could talk about SlideSeq and Visium in a different thing. But at least if you have single cell resolution spatial data, high quality segmentation, shouldn't be able to tell the difference. And in the Harmony paper, one of our examples was harmonizing MERFISH with 10x. What's tricky is that little errors in segmentation, if there's a T cell next to a B cell, and that T cell, because the segmentation wasn't perfect, has 20 extra B cell genes, right? Then it's not going to look like a single cell T cell. It's going to look like a doublet, sort of like a 80-20 doublet.
Okay.
And so it's not going to map perfectly. And so this is what we see sometimes when, if in spatial transcriptomics, we don't always see discrete clusters. And when we harmonize or symphonize, there is, we assume there's a one-to-one connection, one-to-one relationship between cells and two data sets. But if that's not true, because the cells are considerably more mixed, considerably more noisy in one data set, it could lead to some funky behaviors. Yeah, so it's a grain of salt to keep in mind. Yeah.
Got it. Great. If we don't have any other question, thank you everyone. Thank you, Ilya. A really nice talk. We really appreciate that. Thank you so much.
All right. Yeah, thanks for the invitation. Thanks for the questions.
Great.